Okay, hello everyone. How's it going? Good, good. Pleased to see you all here. Okay, today's the fourth lecture in the series that I'm giving, and we've finally got on to looking at translation. So we've looked at DNA replication, we've looked at transcription in prokaryotes, and then we looked at transcription in eukaryotes and RNA processing in eukaryotes. So now we're going to move on to look at translation, which is the second half, if you like, of gene expression. Okay, so the first section of work is taken from chapter 17, which covers the genetic code. So we'll just quickly whip through the genetic code, and then we'll look at translation. Okay. So if you're to look at these nucleic acids, these polymers of nucleic acids we've been looking at, there's and then if you also look at a protein string, like a primary structure of a protein, and you look at the things that make up nucleic acids and the amino acids that make up proteins, there's very little chemical similarity between those different sets of molecules. Okay, so there's no obvious sort of chemical relationship between amino acids and nucleotide bases. But somehow there has to be a link between the two, otherwise we wouldn't be able to make a protein sequence from a nucleic acid sequence. And there's no obvious reason to think that a given amino acid should be matched to a particular nucleotide. Okay, so we'll explain how this happens. So effectively, what we're talking about is the genetic code, which is the relationship between these nucleotide bases and amino acids. And the genetic code is effectively the same in all organisms from all different archaea, or whatever the term is. And so effectively we have one genetic code for all forms of life. And this is kind of another example of how we're now using molecular evidence to explain, to understand evolution. And this sort of comment here from the textbook harks back to the days where, to study evolution, people got tape measured and measured skull diameters and size of the eye sockets and the length of this, and they kind of just measured bones and tried to work out relationships from those things. And it doesn't really make a lot of sense, because a couple of mutations in a genome can change the length of bones and all that sort of stuff. So it's kind of like a so it's much better to understand and use molecular detail to track evolution, because evolution is the development of the DNA genome and the mutations that accumulate in the DNA. That's what evolution is really driven by. Okay. So what is the genetic code? And if you were like a 1950s scientist trying to understand how you relate a nucleic acid sequence to a protein sequence, and you've got no idea, then you start to sort of scribble down ideas on napkins at conferences and try and work out what's going on. And if you just think about the problem, then if you look at a nucleic acid sequence, a DNA or an RNA sequence, you've got a repeating sequence of 4 nucleotide bases. Okay? But when you look at a protein sequence, you've got a repeating sequence of 20 amino acids. So there's quite a big distinction between the number of bases in nucleic acids and the number of amino acid residues in proteins. Okay? You've only got 4 bases, but you've got 20 amino acids. Okay? So clearly, more than one base has to code for, or have information to select. So you need to have, if a single nucleotide base coded for an amino acid, then you'd only be able to code for 4 amino acids. Okay? If a pair of nucleotide bases, in other words 4 times 4, because you've got the first position an A and the second position a T, so that's one combination. You can actually make up 16 combinations of nucleotide pairs. So if you take a pair of nucleotides, you can code for 16 amino acid residues, which isn't enough to code for 20 amino acids. But then suddenly, if you take 3 nucleotides, which is 4 times 4 times 4, because you multiply by the third position, suddenly you get 64 combinations. So there's no clear relationship between the 4 nucleotides and the 20 amino acids, but it's kind of clear that you need to have at least 3 nucleotide bases which combine to make a choice of more than 20. Okay, so if you take 3 nucleotide bases and call that a codon, which is this hypothetical relationship between the nucleotide and the amino acid, then you need at least 3 nucleotide bases to begin to have a code to code for these 20 amino acids. So Francis Crick, famous for Watson-Crick DNA structure, he hypothesized after the DNA structure was published that there had to be an adaptor molecule that is this link between these 3 nucleotide bases and an amino acid. So shown at the bottom here is a messenger RNA that's going to be translated into a protein, and somehow these nucleotides have to impart information to select these amino acids. So somewhere between the messenger RNA and the amino acid, there had to be this adaptor molecule. They didn't know what it was, so it was called an adaptor molecule. It could have been sugars, it could have been proteins, could have been DNA, RNA, it could have been anything. It was an adaptor molecule. It was required to make this link. So people knew this had to happen, and within this adaptor molecule, odds on, it recognized at least 3 nucleotide bases, because only by recognizing 3 nucleotide bases can you be selective for at least 20 amino acids, which is what you need to be selective for. Okay? So that's the kind of the groundwork and the thinking that where people were at in the mid-50s before they actually did the experiments to actually work out what was going on. Okay? So this is the strong hypotheses, this is the smart thinking, which then allowed the experiments to proceed. So what they identified was that this adaptor molecule that they were talking about, it turns out the adaptor molecule is an RNA molecule. All right, now we've talked about different types of RNA molecules in the previous lectures. We've talked about messenger RNA molecules, we've talked about ribosomal RNA molecules, we've talked about tRNA molecules, these transfer RNA molecules, small nuclear, micro, there's lots and lots of classes of RNA molecules, but this is the tRNA molecule, a transfer RNA molecule, and this is the adaptor molecule, or one of the adaptor molecules that can translate from a messenger RNA to select an amino acid residue. Okay, so the adaptor molecule is an RNA molecule called a transfer RNA. This is showing so transfer RNAs are short, single-stranded RNA molecules, and they're coded for by RNA polymerase 3. I think we talked about them in that context. So this is just drawing one of these transfer RNAs, which might be less than 100 nucleotides long, and its primary structure, its single-stranded species, can fold up into a defined structure. So it's got a defined 3-dimensional structure. And this is just showing the 2-dimensional structure, if you like, as you draw it, and it just shows these regions of base complementarity within the single-stranded molecule that allow it to fold up into only one species. It can only form up into this structure. Okay, if you look at the sort of the hydrogen base pairing, complementary base pairing that exists for this molecule, you only get one solution, and it's this fold. Okay, so this is referred to as a cloverleaf structure, and the key points are that in the lower region here, there's a run of 3 nucleotides, and it's those 3 nucleotides that are specifically base paired to the messenger RNA. So this is where the specificity comes to select one of these transfer RNAs is specific for a codon in the messenger RNA. And it's got these other arms which are useful because it needs to recognize a protein, and then it's got this other arm up here which is called the amino acid arm. So clearly that's where the amino acid interaction takes place. This is where an amino acid is attached to the transfer RNA. And then and it's on the 3' end that there's a CCA conserved sort of run here, and it's at this A residue at the 3' end that this AA residue, this amino acid, is attached. Okay, and that's what we're going to be looking at. So there has to be an adaptor molecule that can recognize one of these 64 combinations of this trinucleotide, and this is an example of one of these molecules here. and it's made up of RNA, and it's called a transfer RNA. This is the tertiary structure of the same molecule. Okay, so it's not new, it's just the same molecule now with a structure derived from probably NMR or X-ray crystallography. And again, here's that 3' end where the amino acid is attached. And if you follow the strand, it goes all the way down here, loops around here, and then it comes back up here, and then loops around here, and you end up back at the 5' end. Okay, so it's the same clover leaf structure, but just as a three-dimensional structure. And again, you've got this anticodon arm, and it's the anticodon that's going to bind to a codon in the messenger RNA, and it's at the 3' end where the amino acid will be attached. And at the other domains, the other structure here is important because it has to bind to an enzyme during the process of its function. So we now know that this adaptor molecule is actually a transfer RNA, as shown here. So we have the messenger RNA. It was transcribed from this point here, and then as you read into the sequence, at some point is the first codon that will be translated. So translation doesn't begin at the same point where transcription began. Translation begins a bit further in the RNA molecule, and those parts of this molecule here are important for determining the start site. So the start codon shown here is AUG. The start codon is always AUG, so that's just not a random sequence. That is actually the sequence in the messenger RNA which codes for the first amino acid residue. Okay? And the first amino acid residue of proteins, would you believe it's always methionine? Kind of seems strange, but there's always the same codon, and therefore we always start proteins with the same amino acid residue. Okay? And this is just showing that there's some complementary base pairing between part of the transfer RNA in the anticodon and this codon here, these 3 nucleotide bases. And there are 64 combinations of this triple nucleotide, and there are 20 amino acid residues that can be chosen. Okay? So the strands are always oriented anti-parallel. That's always the case for nucleic acids, and it's no different here. You have 5' to 3' messenger RNA, 3 to 5 anticodon base pairing, and the codon is numbered 1, 2, 3 in the 5' to 3' direction. The anticodon is numbered in the 5' to 3' direction also, but the numbers are in the opposite direction here. So the first position in the codon is the third position in the anticodon, because each is written 5' to 3', and each is numbered 1, 2, 3, 1, 2, 3 in the 5' 3' direction. All right. So transfer RNAs have a similar structure. Okay, there are differences between all of them, but they basically fold up into this cloverleaf structure, which forms that three-dimensional structure that we looked at, with the coding, with those different arms, and with the 3' bit where the amino acid will attach. They're all between 73 and 93 nucleotides long. They all have intramolecular base pairing to form this common three-dimensional structure. They all have a 3' terminus which ends in the bases CCA, and it's the A residue at the end where the amino acid will be attached. As it says here, the 3' terminal A residue is the nucleotide to which the amino acid will become attached. Okay. So we have a situation now where we have 64 codons, 20 amino acids. So clearly more than one codon codes for the same amino acid, because that's referred to as a degenerate genetic code. Okay, not like a degenerate teenager. It's not a naughty genetic code. It's just degenerate, meaning more than one. Sorry, it's a really bad joke. I apologize. But more than one of the codons can code for the same amino acid residue. So multiple codons encode the same amino acid. So we have 64 codons. All 64 codons have a meaning. Every one of those possible codons codes for something. Okay? 61 of those codons code for amino acid residues. 3 of those codons don't code for amino acid residues, but they code for what's referred to a stop codon. And the stop codon doesn't bring in a transfer RNA, but it brings in this other thing, which is a protein, which terminates translation. So when you have one of these special codons here, then that's a stop codon, and that terminates translation. So out of the 61 codons coding for 20 amino acids, there are instances where one codon codes for one amino acid, but that's fairly rare. So there's one codon only for tryptophan. Okay? But there are instances where 6 different codons of these 61, 6 of them all code for the same amino acid, which is arginine. Okay? And typically 3 or 4 will code for an amino acid. All right? So, and then we have a table here which evolved over 5-10 years worth of research of looking at which codon codes for which amino acid, and they did lots of experiments, and it's described in the textbook, and I think it's really quite interesting, but I'm not going to cover it here. But so here's a matrix, the matrix of life, as opposed to the matrix of the movie, which was all about this. So if you look here, each of these there are 64 codons in this table here. The first residue, codon position 1, is A, for argument's sake. So all of these have A in the first position, and then the second position here is determined by the columns. So all of these have U or C or A as the second position, and then the third position is just either of the 4 bases. So you have 4 entries. So, and just through this table, there's a matrix of 64, a table of 64 codons. Here are the 3 stop codons, which don't code for amino acid residues, they code for the end of termination. And here's our special start codon. And there's only one of those that code for methionine. And it's a special codon. It codes for the first residue, or if there's a methionine later on in the sequence. Okay? And then we've just got examples of all of these 4 codons are degenerate, and all of those 4 codons code for leucine, as do these 2 codons here as well. So we have 6 codons for leucine, and we have 2 codons for tyrosine, etc. All right, so it's just the other point of interest, which you may have noticed or may not have noticed, if you look at the third position here, it's always written UCAG, so it's always written in the same format here. Now, it's always the top 2 code for one amino acid, and the bottom 2 code are different. And it's, again, the top 2 code for amino acid, and then the bottom 2 are different. Top 2, bottom 2. Top 2, bottom 2. So if you've got A and G, adenine and guanine, are both purines. Okay? So the third position is the same type of base. So remember, if you look at your nucleotide bases that we would have looked at in proteins and genes, then purines are the large, bulky bases with 2 rings, and pyrimidines are the single rings. I think I've got my purines and pyrimidines the right way around. Check the textbook on that. And also, it makes sense that either the purines or the pyrimidines code for the same amino acid residue, because structurally they're very similar. Okay, so that's the genetic code. The first 2 nucleotides are very important in determining the choice of amino acid residue. The third nucleotide isn't so important in a lot of cases, where the third nucleotide, it can be either of the 4 nucleotides, and it still codes for the same amino acid. And that's the degeneracy in the genetic code. And it's different for each amino acid. So several different codons may specify for a single amino acid. The first 2 nucleotides of each codon are the primary determinants of specificity, which is what I was saying. And the difference in this degenerate code comes at the third position in the codon. Okay? And an example here, alanine is specified by GCU, GCC, GCA, or GCG. So all possible combinations beginning in GC code for alanine. Okay? There are also there's a whole bunch of amino acids, as I showed you, which are specified by 2 codons, and the third of these, at the third position in these codons, is either a purine or a pyrimidine. So that's what I was saying before. It's the third position is either a purine or a pyrimidine. So it's here. So the third position here is either typically of the same type. Okay, so functionally, it's quite well understood what the genetic code is, and so we have a situation there where we have these transfer RNAs recognizing the codons, and we know that the first 2 amino bases in the codon are really important, and the third one is less important. So this is just showing here the messenger RNA sequence on the bottom, and this is just showing GC, GC as the first 2 codons, and either A or G as the third codon. Okay? And you have one transfer RNA bringing in the same amino acid residue that recognizes either of those 2 sequences in the messenger RNA. Okay? And what we say that the third base is where there's a bit of wobble. There's a little bit of freedom in that third base. There's a bit of movement, so you can structurally fit in this non-canonical base pairing at the third base. Okay, so typically A would base pair with T or U in RNA molecules, and here you've got a G not base pairing with a C, but base pairing with effectively a T or a U. Okay, so it's that non-canonical base pairing, but there's a bit of wobble in that codon, and it's only 3 nucleotides, and that's where the degeneracy occurs. So there are only 32 transfer RNA molecules in total, which can recognize 64 or 61 codons. Sorry. So 32 transfer RNAs recognize 61 codons, and out of those 32 transfer RNAs, they attach to one of 20 amino acid residues. Okay? So there's a specific transfer RNA for each amino acid residue, and sometimes there's more than one transfer RNA for an amino acid residue because of the degeneracy in the genetic code. So this is just showing, again, this wobble concept in a bit more detail. So at the third base pair here, typically you'd expect an A to base pair with a U, because that's canonical Watson-Crick base pairing. That's what we always see. And then you get the nice, neat base pairing at the position here. In this instance here, we've got the GC, but rather than an A, we've got GCG, but it's still bringing in the same transfer RNA, and there's a bit of, and you get this kind of wobble base pair, which you don't get in long strands of nucleic acid, like in DNA and in transcription. You don't see this sort of wobble. You always get the specific base pairing, but in the relationship between the transfer RNA and the messenger RNA, you can get this non-canonical base pairing at the third place, and it's called a wobble base pair. And this is just showing a G, rather than base pairing to a C, you can base pair to a U. So effectively, the genetic code here for alanine varies on the third position, and they can all bring in the transfer RNA. So the first 2 bases of the messenger RNA codon always form Watson-Crick base pairing, and it's these 2 that give the most specificity in the genetic code. Okay, so the other thing you've got to think about when you're looking at a messenger RNA and how it codes for a protein is that you've got 3 bases that make up the code, but are those 3, is the codon positions 1, 2, and 3, or is it positions 2, 3, and 4, or is it positions 3, 4, and 5? So that you can have a sliding window of one base pair, which can group 3 nucleotides into 3 possible reading frames. And this is just a slide that tries to explain that concept. So this is a messenger RNA shown here, and I've just drawn these squares around the messenger RNA, showing that A, U, and G are a codon. Okay? If you slide that reading frame, so you slide it 1 nucleotide to the right, like that, and then you look at the same sequence of bases, but you interpret these groupings now, down below here, as codons. Okay? So we've still got the AUG. We've still got AUG, but now we're starting at the second base, and we're grouping UGG as a codon. Okay? And that's called the second reading frame. All right? And then you can do the same process again. You can slide it one nucleotide to the right along the same messenger RNA to get the third reading frame. And then when you interpret, so there's three ways of reading one messenger RNA. Okay? So again, when you don't know how the genetic code works and how translation works, you have this foresight that, well, does each base pair, do you slide one along? So if I was to read this messenger RNA, is the sequence going to, for my protein, going to be met, val, arg, blah, 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 or is it going to go met, trip, glyve, val, cis, alla? Because do you read nucleotides in groups of three, or do you slide along three at a time, or do you slide along one at a time? Okay, these were concepts which you take for granted that you slide along three at a time. I think you take that for granted, I don't know, but it wasn't perceived, it wasn't known in the 1950s, so they had to do experiments to work this out. Okay? So we have three possible reading frames, and each of those reading frames would give you a completely different amino acid sequence. Okay? So there's no similarity between either of these three amino acid sequences based on one messenger RNA. So if I'm looking at a messenger RNA sequence on a computer and someone says, what's the protein sequence? Well, you're going to scratch your head and go, well, I don't know. It depends where the reading frame is. Do I start this base, or this base? Okay? So the cell has to have a specific way of starting translation at a particular codon to read the right protein sequence. Otherwise, you'll get two possible alternate sequences, which are nonsense. Okay? Only one of those sequences will code for a functional protein that plays a role in cell biology. Okay, so as I said before, we've got what's called a start codon, which is the ATG, which codes for methionine. So DNA is ATG, RNA is AUG. So that helps determine the start amino acid and also the correct reading frame. Okay, and then that will be read through until you get to the stop codon, which is one of those three stop codons. And between the two of those codons is an intervening sequence, which is the reading frame, which is converted into a protein, and there are no stop codons in this sequence here, and it's called an open reading frame. Now, if you do the maths, again, on a napkin at a conference, you're having a chat, and you'd say, well, the chance of there being one of these three stop codons, so 3 out of 60, if you like, the 61 codons, no, the 64 codons, 3 of them are stop codons. So about 1 in 20 codons, by chance, would be a stop codon. Which means if you were to look at a sequence just by chance alone, you'd expect an average reading frame to be 20 nucleotides long before the stop, not 20 nucleotides, 20 codons long. So if 3 of 64 codons are stop codons, then randomly you'd expect there to be one stop codon every 20 codons. But open reading frames are often easily over 300 codons long. Okay? So you can tell when you see a long stretch of sequence without a stop codon in that particular reading frame, then that's going to be a gene. And this is some of the bioinformatics tricks we use nowadays, because we're coding hundreds of organisms daily. People are going around getting bacteria, and their job is just sequencing them. And then you use bioinformatics to work out where all the genes are in the gene sequence, and then predict the proteins, and then you do homology to predict the folding and function of proteins. And people are just churning through organisms at a rapid rate to determine, find things in these, like mining these sequences. So this is some of the rudimentary bioinformatics they do in terms of trying to identify where a gene is. When you don't know what the protein product is, you don't know where the promoter is, and you can use some of these basic techniques. So in the early days, in the 50s, they weren't sure whether codons overlapped or didn't overlap. Okay, so we know this to be true, that codons don't overlap, but they had to prove that codons didn't overlap. So each codon is an independent unit here, and what they effectively did, they did mutational studies where they'd change one nucleotide in a messenger RNA and look at the protein product. And if that one nucleotide change caused one amino acid change, then it meant it was a non-overlapping sequence. Whereas if that one nucleotide change caused 3 amino acids in a protein to change, then it would have been a non-overlapping genetic code. Okay? So they did these kind of experiments and determined that a single base pair changed one amino acid, and therefore it was a non-overlapping genetic code. Okay? And then here's some of the experiments they did. They did insertion experiments, which changed all of the amino acid sequence beyond the site of that insertion. They did deletion experiments, and that changed the amino acid sequence for all following amino acids, or all, beyond that change. They did an insertion and a deletion, so you had a different sequence, but the deletion put it back into the same reading frame, so that the protein sequence returned to the wild-type protein sequence. And then they did a triple insertion, and that just inserted one amino acid residue. So once they did these experiments here, it was beyond doubt that it was non-overlapping. There was no argument anymore. It was solved. Okay? So I know we always use mutational experiments to manipulate protein sequences so we can, do all these studies. But in the early days, they did mutational studies to actually understand the process, the genetic code. Okay, so that's all I have to say about the genetic code. I think you guys have an understanding of the genetic code beforehand anyway, but hopefully I've just solidified and maybe extended your knowledge on the genetic code. So now we're going to look at protein synthesis, otherwise known as translation, which is the second half of gene expression. So transcription followed by translation gives us our proteins. So with all of these processes of DNA replication and transcription, I talked about this polymerization process having an initiation process, an elongation process, and a termination process. Okay, and the same is true for translation. We have an initiation, elongation, and termination phase. During the initiation and elongation phase, other stuff has to happen in the cytosol. We need to get the amino acids and effectively attach them to transfer RNAs. So that happens in this, that also has to happen as part of the initiation and elongation. We have to have the amino acids become activated and attached, get the transfer RNA charged. And also, we're not looking at protein folding, but once we put these amino acids together into a long string, there has to be a process of folding to give you a functional protein. Okay? So in terms of gene expression, you'd have to fold the protein as well before you have an active protein. So we're looking now at the process of reading the messenger RNA and bringing in these transfer RNAs and amino acids, and then bringing in a second one to then join these two amino acids together, then bringing in another one, and then join that together. So this process of bringing in these transfer RNAs and sequentially moving along the messenger RNA and then building a protein sequence. So this is just showing one of these 32 transfer RNAs attached to one of our 20 amino acids, and then you've got Watson-Crick base pairing here. Okay? So each transfer RNA only is bound to a single amino acid. There's only one amino acid type that can bind to one specific transfer RNA. It's a very specific interaction, but it turns out there were 32 of these in total, and they bind to one of 20 amino acids. So what is the big molecular machine, the big thing that carries out translation? Well, it's something called a ribosome. Okay? So we've got, like I said before, we've got proteomes, ribosomes, exosomes, spliceosomes, all these things ending in -ome, which instantly tell you it's a big complex of multiple things that come together in a specific way to form this big molecular machine. Okay? So we've got this thing called a ribosome. And the structure of a ribosome facilitates protein synthesis by bringing together the messenger RNA and the particular codon in a messenger RNA and the corresponding transfer RNA, which has an amino acid attached, and then the ribosome then facilitates the catalytic process of forming a peptide bond between the two amino acid residues. Okay? So these two halves, these big circular bits here, these two halves here are the small subunit and the large subunit of this thing called a ribosome. Okay, so this is a little cartoon here. And these three things here are domains, if you like, of the ribosome. And what you can see here, we've got three domains called an A, P, and an E domain. I'll describe them more later. Each domain spans between the large subunit and the small subunit. So neither subunit in isolation is functional, but when you put them together, it forms these three pockets, these three domains, and then the subunit, the entire ribosome can form its function. Okay? So a ribosome is made up of two halves, and these three sites here span both halves. And we have a small subunit which binds to the messenger RNA, and a large subunit which binds to the small subunit. In the small subunit is what we call the decoding center. So in other words, this is where the messenger RNA is bound and where the transfer RNA will base pair to the messenger RNA, and that's called decoding, and that's where the decoding takes place. And in the large subunit is where the peptide bond formation takes place. Okay? So even though these three domains are shared between the two ribosomes, you have the small subunit where the base pairing occurs between the nucleic acids, and in the large subunit is where the peptide bond is formed between the amino acids. So what are ribosomes? We have two subunits, the small and the large. They're made up of, the large subunit is made up of 36 proteins, 33 of them being different proteins, with a couple of them occurring twice. So you've got 36 proteins and two ribosomal RNAs. In the small subunit, you've got 21 different proteins and one RNA molecule. Okay? So I talked to you about the ribosomal RNAs being transcribed by RNA polymerase 1, and I was telling you they're not translated. Okay? So these rRNA molecules are functional RNA molecules. They're not translated like a messenger RNA is translated. These ribosomal RNAs make up these things called ribosomes, and they form these complexes with many proteins to form these complexes. Okay? Now, when you look at a ribosome and what a ribosome does, all of those important functions that a ribosome does are catalyzed by RNA, not protein. Okay, so it's another one of these proteins where RNAs carry out all of the functions. So RNA is responsible for binding to the transfer RNA. The RNA is responsible for forming the peptide bond, and the RNA is important for forming the connections between the two subunits. So the proteins are obviously carrying out functions, but they're more structural functions, and maybe some other functions will be ascribed to proteins over time. So this is showing the structure of a ribosome, both halves, and it's a complex structure. The textbook says it's more complex than a DNA polymerase. So here's the bottom half, the small subunit, and here's the top half, which is the top subunit. And it's just color-coded a few of these proteins. And there's a cleft here where the transfer RNAs will be brought in. Okay, so we're not showing the messenger RNA in this diagram, and we're not showing the transfer RNAs in this diagram. We're just showing the two halves of the ribosome. This is the eukaryotic ribosome. It's bigger, it's made up of more proteins, but it's functionally and structurally very similar to what we see in eukaryotes. Again, it's one of these fundamentally conserved processes. Again, you've got the small subunit and a large subunit. Yeah, there's a few more proteins involved, but again, you've got very similar structure. This is a scanning electron micrograph, which is really fascinating. This would have been, very hot research paper in its day. What it's showing here, you can actually see the DNA molecule shown here, and then during the DNA is being transcribed, which is these RNA molecules coming off the DNA molecule, and then those RNA molecules are being translated whilst they're being transcribed. Remember, it's coupled in bacteria. So this polymerase has only made a short messenger RNA, and a couple of ribosomes have attached to that short thing there. As the polymerase is working down the DNA, it's making a long messenger RNA molecule, okay? And lots of ribosomes have attached. And then you can't see it here, but there would be a long polypeptide string from this one and a short polypeptide string from that one. So they've really captured the process of transcription, translation in an electron micrograph here. And it really informs on how the process works. So in that sense, it's really an amazing electron micrograph. Okay, this is cartoon form, just showing that the RNA polymerase is reading along the DNA, and there's the bubble there. It's making this messenger RNA, which ribosomes are attaching and making these pink polypeptide strings, which are going to be the proteins. Okay. So a ribosome is said to be a ribozyme, meaning the enzyme function of the ribosome is predominantly through the RNA molecules embedded in the ribosome. Okay, so the ribosome is a ribozyme. The ribosome can be inactivated by making base pair changes to the RNA sequences within the ribosome, which tell you straight away that when you can knock out the enzyme activity by making base pair changes to the RNA, then those RNA sequences are really important. Okay, and you can also knock off the proteins in the ribosome and still maintain the function of the ribosome, telling you that the proteins aren't involved in the catalytic activity of the ribosome. Okay. So I showed you that when the two subunits of the ribosome come together, they form this A site, P site, and E site, which straddle the two halves of the ribosome. What do these letters refer to? The A site, excuse me, is the location where the transfer RNA, which is attached to an amino acid, comes into the ribosome. The P site is the location of where the transfer RNA will be during that is attached to the poly to many amino acid residues. So where the nascent protein is being made, that will be at the P site, and the E site is where the transfer RNA, once it's lost its amino acid, will exit the molecule. It'll be a lot easier to show. Have I got them in a slide? Yeah, here. So here's those three sites here. You've got the exit site where the transfer RNA exits. So it comes in here, and then as the ribosome shuffles along, the transfer RNA gets moved between these three sites, and then it will exit from the exit site. So I'll show that in some animations coming up. So we have three sites: A site, P site, E site. The A site is named because it's the location of the Aminoacyl-tRNA, so the transfer RNA with the amino acid attached. P site is the growing polypeptide chain attached to a transfer RNA, and the E site is where the transfer RNA without an amino acid exits. So each transfer RNA starts in this A site, and then as the ribosome shuffles along the messenger RNA, that transfer RNA is moved from the A site to the P site to the E site. So that's the sites I was talking about. Like I said before, the large subunit carries out peptide bond formation, and in the A site here will be the incoming transfer RNA, which has an amino acid attached to it. In the P site will be where the growing polypeptide chain will be, and the E site will be where the transfer RNA exits. And again, this is the same sort of structure I showed you earlier, but now it's showing in green here, if you look carefully, it's showing three green blobs. That's 3 transfer RNAs within the ribosome. And these purple bits here are the 3' terminus where the amino acid is going to be attached. Okay? So you can see here, these 2 amino acid residues, had they been in place here, are brought into close proximity within the enzyme. And then when they're brought into close proximity, somewhere in here there'll be some RNA strand which can facilitate the peptide bond formation between those 2 amino acids. Okay? So again, you can't separate structure and function when you look at these 3-dimensional structures. They start to reveal the secrets of how these enzymes carry out their function. Okay? And it's all related to structure. Okay? And also, this little red bit you can't see in here, you've got to imagine that along here is the messenger RNA. And it just shows the messenger RNA is located down here, where you're getting the base pairing between the transfer RNA and the messenger RNA is taking place here. Okay? So this is the decoding site of the small subunit here, and this is the peptidal transferase site of the large molecule, of the large subunit. And between those 2 subunits are these sites where these transfer RNAs are bound. So neither subunit can work in isolation. The whole thing has to assemble for it to become functional. This is now, so in the early slide I showed you the ribosome by itself without the transfer RNAs. Now we're showing the ribosome with the transfer RNAs, and now we're going to take away the ribosome and just look at the transfer RNAs and the messenger RNA. So within that molecule, this is the structure we see. We see the messenger RNA inside the small subunit, and the messenger RNA is wound or bent to give this structure here. Okay? So as the ribosome slides along the messenger RNA, it forms a local structure of that messenger RNA. It bends it to form this kink. Okay? And then this is the position of the transfer RNAs, showing the codon-anticodon base pairing here. So here's the base pairing between the transfer RNA and the bases of the messenger RNA. If you swivel this around, you just take the whole thing and swivel it around, then you're looking at the same thing here, just from a different angle. And this is showing how the kink here causes these transfer RNA molecules to come close together to bring these amino acids residues together. And then you can then form a peptide bond between these things that are brought close together in space. Okay? So again, it's really great sort of understanding of what's happening within these molecules at a molecular level. So, like we said, ribosomes, 2 unequal subunits with the peptide bond being formed in the large subunit, and the catalytic activity is a peptidal transferase center, is what it's referred to, and that's the enzyme activity that we're seeing here. And on the small subunit, you've got that base pairing between the transfer RNA and the kinked messenger RNA, and that's said to be the decoding site on the small subunit. And as you can see here, we have 2 transfer RNAs, each with an amino acid attached, and then the peptide bond will be formed within the ribosome. This is this sort of cyclic diagram showing initiation, elongation, termination of one round of translation. So we start down here. You've got a messenger RNA with a start codon, stop codon, and then the small subunit attaches to the 5' end of the messenger RNA and somehow recognizes the first AUG. Okay? Now, the transfer RNA, the first one brought in, is the one that binds methionine. Okay? So that recognizes AUG, and then once that's in place, once the small subunit has bound to the messenger RNA, and then the first transfer RNA is brought into place, that's when the large subunit then binds to the small subunit. Okay? So this has to happen first during initiation. And then the large subunit comes in, forms this complete process here, and then another residue comes in, and then you get this bond formation. And then the ribosome effectively clunks along the messenger RNA, and it works through until it comes to a termination sequence, which brings in not a transfer RNA, but a this other thing which just adds water on the end, and the peptide leaves. And during the process of walking along the messenger RNA here, one codon at a time, it's brought in one amino acid residue at a time, and it's joined them together one peptide bond at a time to make a polypeptide. And then it comes apart and then can reassemble and do the process again. So it's a cyclic process, and the messenger RNA will be translated as many times as it can before it's degraded by the exosome, which we talked about last week, the exosome that degrades the messenger RNAs. Okay, so to continue where I left off, we just talked about this cycle of translation, and some of the key points which I might have mentioned already was that when you look at the three-dimensional structure of these complexes, you can see that there's a kink introduced into the messenger RNA, which is important in, and these pockets here are important in bringing these amino acids together close in space, and then as the proteins are being synthesized, there's a channel where the polypeptide exits from the ribosome, and also the messenger RNA needs to be unfolded as it comes into this channel here, so that any tertiary structure is removed, so that the tRNA can base pair with the messenger RNA. And as the polypeptide exits this polypeptide sort of tunnel here, it needs to be folded prior to being an active protein. So now we're going to look at some of the process of an amino acid needs to be chemically bonded through a covalent bond to a transfer RNA. And then that transfer RNA can take that amino acid into the site where protein synthesis occurs. So there's a side issue that we need to look at, which is the activation of these amino acids. So this is what we're talking about here. So each amino acid is specifically bonded to a particular transfer RNA. Okay? And we say this is the charging of the transfer RNA. Is the attachment of this amino acid. So a charged transfer RNA has an amino acid attached to it. Okay? And the way the amino acid is attached, it's through this acyl linkage between the carboxy group of an amino acid and the 2' or the 3' hydroxy end of the transfer RNA. Okay? So we've been talking about the 3' end of the transfer RNA, but actually the 2' hydroxy is also exposed at the 3' end. So we could refer to it as a 2' end or a 3' end. Okay? We've got the 5' end and the 3' end, but at the 3' end of the transfer RNA, there's 2 free hydroxy groups, and you'll see that in a diagram. So there's an enzyme involved in attaching the amino acid to the transfer RNA, and that's called a tRNA synthetase. Its full name would be an Aminoacyl-tRNA synthetase. Okay, so there's a whole bunch of these synthetases, and there's a distinct amino acid tRNA synthetase that's responsible for the attachment of each of the 20 amino acids. Okay, so there's at least 20 of these Aminoacyl-tRNA synthetases. Okay, and they're responsible for attaching an amino acid to a transfer RNA. Okay, and it's a two-step reaction. It's not a single-step reaction. So the first step in the reaction is the activation of the amino acid. So the amino acid is reacted with an ATP molecule to form a high-energy bond, and that's the 5-aminoacyl AMP. So so you get the amino acid being covalently bonded to this nucleotide, and then that then is attached to a transfer RNA, and then we say that the transfer RNA has been charged because it's got the amino acid attached. So we've got this two-step procedure which is catalyzed by the Aminoacyl-tRNA synthetase. And we've got 20 Aminoacyl-tRNA synthetases involved in adding 20 amino acids onto these different transfer RNAs. So this is looking at the first step in the reaction, which is taking an amino acid, shown in reddy pink here, and that amino acid is reacted with a nucleotide, so an ATP, and the carboxy terminal of an amino acid. So just have a think about this blob here. You've got the amine group, the carboxy group, and then you've got the alpha carbon, and then you have the side chain of the amino acid here. So we've got 20 different amino acids with 20 different R groups, but every single amino acid has the amine group and the carboxy group, obviously. Okay, you should be clear on that. Every amino acid has this carboxy group here. Okay, and it's the R group that is either, if it was a H, it would be a glycine. If it was CH3, it would be alanine. If it was, tyrosine, it would be an aromatic ring with an OH on it. You know all the different structures. It would be 20 amino acids. There's the side chains here, but here's the carboxy group that all amino acids have, and that carboxy group is attached to the alpha phosphate here to give you this structure here. So this is an amino acid, which is now referred to as an aminoacyl AMP, because the amino acid is attached to the AMP molecule. So the ATP is cleaved, bonded to form this. Okay, and then this is attached to the transfer RNA. Okay, so this is a high energy bond which is easily broken and then attached. So we're talking about that this is the transfer RNA here now. So we know that the last residue of the transfer RNA is always an adenine, okay, because it's conserved through all the different transfer RNAs. They all have the same, I think it's CCA is the 3' end. So at the 3' end here, it's also got a free 2' end. Okay, it's the same, this sugar has both of its OHs free. So the amino acid can be attached to either the 2' or the 3' OH. Okay, and then, so this is showing the attachment at the 2' here, the attachment at the 3', so either of these cases occur, but there's a transesterification that happens, so you end up with the amino acid attached to the 3' OH, regardless of which aminoacyl transfer RNA and to which of these free OH groups. Doesn't really matter which it is, it ends up at the 3'. Okay? And that's what goes into the ribosome, is this charged transfer RNA with an amino acid attached to it, okay, on the 3' OH. Okay? But there's a bit of subtlety in the attachment of this amino acid, depending on which aminoacyl-tRNA synthetase is doing the job. So we've got, if you think about what's happening, you've got a transfer RNA with its anticodon going to bind to the codon. Okay? So regardless of what amino acid is attached, you're going to have that anticodon recognition is going to occur, because it's in a different part of the transfer RNA to where the amino acid is. So if the wrong amino acid is attached, then the wrong amino acid will be put into the protein. Okay? So it's very important that the right amino acid is attached to the right transfer RNA, because whatever's put there is going to be added to the protein based on that codon-anticodon recognition. So people are referring to this aminoacyl-tRNA synthetase. The way it recognizes a transfer RNA is said to be the second genetic code, because it plays a vital role in determining that specificity has to be correct, because whatever amino acid is attached will then be added to the protein when the anticodon-codon base pairs. So this is just showing the protein here is shown in the big thing, this green molecule here. I've shown you this earlier. This is the transfer RNA. Okay, and again, here's the transfer RNA. And here's the amino acid shown here. There are two types of tRNA synthetases, and this is the two types shown here. And it's the bonding between these different arms of that transfer RNA with the protein sequence here. It's these different sites of interaction which form a very specific interaction. And then that bonds the right transfer RNA to the right tRNA synthetase so that the amino acid can be attached. And if something goes wrong in this sequence here, then effectively the wrong amino acid will be put into a protein. Okay? So this is, again, one of these highly specific interactions which someone has coined the second genetic code, and it seems to have taken a bit of popularity. So this is just getting back to the cloverleaf structure I showed earlier of a transfer RNA. And if you remember, we had the anticodon sequence here. We have this amino acid arm here with its CCA residue. All transfer RNAs have a CCA. And it's to that A at the 3' terminal end where the amino acid is attached. Okay? But the textbook reminds you that it's also got a 2' hydroxy is also present. And the resultant reaction is that the amino acid is attached to the 3' OH group. And you've got these arms, this D arm, this T arm, and these specific residues on each transfer RNA, and these are really important for recognizing the right tRNA synthetase, the right enzyme that's going to add the amino acid. Okay? So I think the book color codes these bases here. I think the black ones are invariant between different transfer RNAs, and the ones that are color-coded are different between transfer RNAs, and they're involved in binding to the correct tRNA synthetase. And then effectively we get the attachment of the amino acid on the 3' end of the tRNA. Again, whenever we're looking at these polymerization reactions, we always think about the fidelity of the reaction. The error rate for protein synthesis is about 1 in 10,000, which is significantly higher than we see for DNA replication. But again, we've talked about this in relation to transcription as well, in that any mistakes that are made are transient in the cell, because the cell is going to make multiple proteins from one messenger RNA. So if there's a mistake incorporated, it doesn't have a long-term consequence for the cell. So any defective protein will normally be broken down fairly quickly, and there are good copies of the proteins to carry out the function. All right, so let's just look at the process now of translation itself. So looking at how the ribosome is reading the message and how the transfer RNAs are being brought into the molecule. So we have an initiation phase. Now, I've told you that there are 3 reading frames. In one of those reading frames, there's an AUG, and that is important in helping bringing in the first methionine residue. Okay? And it's the small subunit that recognizes this AUG, and then during this recognition process, the FMET, it's shown here, the initiator transfer RNA, the first transfer RNA, will be the one that recognizes the AUG, and it codes for methionine. Now, the first transfer RNA brings in a modified methionine. Okay? So in eukaryotes, we have a modified methionine which is used to initiate translation. So the initiator, Aminoacyl-tRNA, is brought in here with an FMET attached. Now, once this initiator, once this complex is in place, it's recognized by the large subunit, and then we have this A site is formed because of the large subunit, and also the peptide transferase site is in the large subunit. So incoming transfer RNAs can come here to be bonded to this amino acid here to form a peptide bond. Now, there's this AUG sequence here alone isn't enough to give specificity to start translation. Okay? So there has to be some other sequence in the messenger RNA to give specificity of where this small subunit will bind. And don't forget, within this small subunit, it's got an rRNA molecule in it, and it's the rRNA molecule that's going to bind to the messenger RNA. And it's that base pairing that helps initiate this process. So somewhere in this messenger RNA is a sequence that we call the Shine-Dalgarno sequence. I'm not sure if you're familiar with the Shine-Dalgarno sequence, but it's a conserved sequence in prokaryotes in the 5' end of the messenger RNA. And if you just zoom in here to this 5' end of the messenger RNA, in the Shine-Dalgarno sequence is this AGGA-GGU sequence. And this conserved sequence, which is in many of these messenger RNAs, is recognized by the ribosomal RNA that's within the small subunit. And it's got the complementary sequence to this Shine-Dalgarno sequence. Okay? So you get this base pairing between the messenger RNA and the rRNA that's in the small subunit of the ribosome, and this is positioned just enough base pairs away to put the AUG sequence into this the correct part of the ribosome, okay? Which then allows this transfer RNA to come in to this pocket here and to base pair. So this Shine-Dalgarno sequence is really important. John Shine is an Australian scientist, and he was. I'm not sure if Dalgarno was an Australian or not, but certainly John Shine is an Australian scientist. He's still kicking around. He's in Canberra these days. He was at, when I was doing my honours I was at UNSW, I remember walking down the corridors and you'd see John Shine walking down the corridors and you go, that's the guy. He's the guy from the Shine-Dalgarno sequence. So it was kind of nice to be in the presence of some of these guys and to sort of meet and talk to them and stuff. He's now in Canberra, I think, I'm not sure. If you go to Canberra, there's the Australian Academy of Sciences, where all of the high-ranking professors, they can be inducted. It's like the Hall of Fame in music, or it's like getting a Grammy or getting an Academy Award, is when you're inducted into the Australian Academy of Sciences. And they've got a building, and it's called the Shine Dome. And it's in Canberra, so the building's named after him. So anyway, so prominent scientist involved in recognizing this Shine-Dalgarno sequence, which is really important for understanding how the process of translation, this complex forms in E. coli. And this is just showing the complementary base pairing between the messenger RNA conserved sequence and the sequence that's present in this ribosomal RNA, which is a part of the ribosome. So there's a mistake in the title of your notes, and I've just tried to fix it up here, that we've been looking at the initiation of translation in E. coli, where FMET is brought in. Okay, so there's a modified amino acid is the first methionine. In eukaryotes, it's a normal methionine that is brought in. But what we have in eukaryotes is that we have a distinct transfer RNA that brings in that starts the process, and there's another transfer RNA that brings in your bog standard methionine as a residue in a protein. So there's two transfer RNAs. One is involved in bringing in the initiator methionine, and the other is used for internal methionines within a sequence. Whereas in E. coli, there's a modified methionine that is brought to the initiator codon. So there's a specific amino acid that initiates protein synthesis in prokaryotes, but in eukaryotes, it's not a specific amino acid. It's a standard methionine, and it's a different transfer RNA that's used. Okay, when you look at mitochondria sequences and how mitochondria behave, so the mitochondria's got a genome. The mitochondria codes for genes. transcribes and translates proteins from the mitochondrial genome and the chloroplast genome. And in this situation, the initiation of translation begins with N-formyl methionine in eukaryotic cells because it's what's happening in the mitochondria or the chloroplast. Whereas in the genome, in the nucleus of eukaryotic cells, it's the 2-transfer RNAs bringing in the standard methionine. So again, this is this kind of molecular understanding that really supports the idea that mitochondria and chloroplasts were incorporated into some eukaryotic cell during evolution, because they still have all of these properties of the bacteria. Okay, so when we look at initiation in eukaryotic cells, we have, so we're moving into eukaryotics now. Translation is generally similar between eukaryotes and bacteria cells, and we'll look at that process. Not only do we have ribosomes and messenger RNAs and transfer RNAs, but we're not going to look into the details of this because there's too much going on, but there are also other proteins involved, and it probably doesn't surprise you that there are other proteins involved. Some of these proteins are involved in hydrolyzing GTP and ATP, and don't forget ATP is the energy molecule of the cell. So when you're hydrolyzing ATP, you're basically consuming energy to drive a process forward. So some of these other proteins involved in translation are hydrolyzing ATP to generate energy to drive the process of translation. And these are sort of these EL factors, these elongation factors, and they're involved in translation. And like I said, in bacteria, the ribosomes are recruited to the RNA based on base pairing to the Shine-Dalgarno sequence, which is in the 5' end of the messenger RNA, 8 to 13 nucleotides away from the AUG. In eukaryotes, it's different. In eukaryotes, don't forget, we've got a 5' cap, and that 5' cap is important in recognizing the ribosome. So the ribosome attaches at the 5' end, and then the ribosome scans along the messenger RNA. And when it hits the AUG, that's when initiation of translation begins. So we don't have Shine-Dalgarno sequences in eukaryotes. We have that in bacteria. In eukaryotes, we have 5' cap messenger RNA. It's not mentioned in the slides here, but the ribosome attaches, and then it slides along in the 3' direction until it comes across an AUG. So that's how initiation begins. We then have elongation of the polypeptide chain. So the polypeptide chain is lengthened 1 amino acid at a time. Each amino acid is brought in by a transfer RNA binding in an adjacent site in the ribosome, and then you get base pairing between the transfer RNA and the messenger RNA through that codon-anticodon interaction. So I've tried to do an animation based on the diagrams in the textbook to show what's happening here. So here we have the initiator transfer RNA in place, and then the large subunits come in place, and we have these sites formed, these E, P, and A sites. So the A site is where the aminoacyl transfer RNA will come in. So this is the transfer RNA which has been charged with an amino acid, the process we talked about. So effectively, there are these helper proteins that we don't need to worry about too much, which are bound to these high energy bonds here, and that comes into the site. You get base pairing between the codon-anticodon, and then you've got a very slight pause where once this is recognized, this GTP has to be hydrolyzed for something to happen. And during that slight pause, you can get make sure that the correct tRNA is brought into place, and then effectively you hydrolyze that GTP to a GDP, and then that allows some sort of structural change so that as you hydrolyze that molecule, you get a shift in structure, and those two amino acids are brought into the same proximity. And then in the peptidal transferase catalytic activity here, you can then form your polypeptide bond between two amino acid residues. So So then you get the charging of the next amino acid. So at this site here, let's look at the diagram in the textbook that shows you what's going on during peptide bond formation. So you've got the F-met, which was there in place, and then the first codon specified for this transfer RNA, which brought this amino acid into place. And then, so you've got the F-met here, you've got the new amino acid here, and basically you're going to get a polypeptide bond formed. And hopefully you can remember from proteins and genes, I don't know if it was, I'm sure it was emphasized, you get a polypeptide bond formation through a condensation reaction where you lose water and you get the carboxy group of one amino acid condensing with the amino group of the adjacent amino acid to form a polypeptide bond. Okay? So where you had a carboxy group and an amine group, you now have C double bond O attached to NH, have formed the next amino acid. So that's what's happening here. You've got your carboxy terminal, you've got a carboxy group of one amino acid here, and you've got the amine group of the other amino acid, and you get a condensation reaction between the two. It's the N group attacks the group here, and then that forms this bond, and then you lose these the hydrogens. So effectively, you get the formation of this NH-CO bond here, which is a polypeptide bond between two adjacent amino acids. Okay, so it's fairly straightforward, but you may want to refresh your understanding of what a polypeptide bond is and how it's formed through a condensation reaction, the removal of water between two amino acid residues to give you a polypeptide bond here. And this all occurs in the large subunit of the ribosome at the peptidase transferase site, and this gives you polypeptide bond formation. So as I showed you in that diagram, the amine group here attacks the carboxy group here. So effectively, the alpha amino group of the incoming amino acid acts as a nucleophilic site that attacks and displaces the transfer RNA, and then this attaches to here. So it displaces the transfer RNA and forms a bond to the next amino acid. So effectively, you get the first joining together, the first polypeptide bond, and the start of your protein sequence is now being joined. And then the ribosome translocates one codon, which now exposes 3 new nucleotides here for the next incoming transfer RNA. And then another... So the movement of the ribosome, this translocation of the ribosome, again involves hydrolysis of a GTP molecule by one of these elongation factors, and that just provides energy for the process. So this is just showing one of those other factors coming into play here. Then you get translocation of the ribosome, and then the freeing up of that site. So then the next amino transfer RNA can come in, bringing in the next amino acid residue, so they can form the next polypeptide bond. Okay? As this translocates along one base pair, it moves the transfer RNA that was initially bound to F-met into the exit site, and then that can then leave the molecule. Okay? So you get one coming in here and one leaving there. Translocation, one coming in, one leaving, translocation. You just get this cycle of sort of... Okay, I can't tell you how long it took me to do that animation for a 2-second display in lecture, so I'm going to play it again just because I made it. So effectively, what I'm trying to show here is the ribosome translocating one codon at a time through the messenger RNA, and it shifts these transfer RNAs into different pockets within the ribosome, which then allows these different processes to occur. So you get this peptide bond formation, and then the coming along, and then, okay. All right, so I don't know why I put them in here, but I put in some summary slides which just probably have a bit too much detail in them. But the first step in elongation is the incoming aminoacyl-tRNA binds to the ribosome. We have this hydrolysis of GTP, and then you get this peptide bond formation, and it involves the attack from one amino acid, the amine group, on the carboxy of the other amino acid, forming a polypeptide bond and that occurs in the P site. And then the third step is the translocation of the ribosome, and again, translocation is driven by hydrolysis of GTP. So it's a fairly energy-intensive process, consuming these high energy bonds, which were made through, obviously, sugar molecules being, glycolysis and stuff, producing ATP molecules, which are now being consumed during synthesis of polypeptides. So 3 steps of elongation are repeated as our messenger RNA is read. And then we get to the termination process, which I've just got a quick animation to show that, and then we've finished. So termination of protein synthesis. So we have these other factors which are not transfer RNAs. We have these release factors, these RF factors. And these release factors, as you can guess, they recognize stop codons. Okay? So when the ribosome is translocating along, and when it finally reaches one of these stop codons, which occur fairly infrequently, much less frequently than you would have presumed by chance, once these stop codons are read, there's no transfer RNA that recognize them, but there's these release factors. And these release factors are proteins which kind of mimic transfer RNAs. Okay? And they come into the site where the peptide bond's forming, and rather than another amino acid being added to the growing chain, a water molecule's added to the growing chain. So effectively, you have the messenger RNA being read, and after a couple of hundred codons, you get to this stop codon. There's no transfer RNA that can base pair with UAA, but there's a release factor which can recognize UAA. So this protein comes in here, and effectively it resembles a transfer RNA, and effectively it just adds a water on the end and causes termination of translation. So now you have your fully formed polypeptide chain, and then these subunits will dissociate, and as we saw in that cycle of diagram, can then reassemble to undergo another round of translation. And that's it. So, so thanks for your attention. If you've got any questions about this or any of the other lectures, come and have a chat.